myself Dr. Vishal Deshmukh from Department of Botany, Jagdamba Mahavidyalaya, Achalpur City, District Amravati, Maharashtra. Today I am here to discuss about protoplast culture. 1880 has introduced the term protoplast, and they have been definition that cell without protective cell wall is called as a protoplast. Custer in 1935 in his book Plant Cell used the term gymnoplast for naked plant cell, and Constable in 1982 define it as a living matter enclosed by plant cell wall living matter enclosed by plant cell walls. Cooking 1972 describe it as a naked plant cell. You can find on the left a naked plant cell without any type of cell wall. So if you differentiate between plant and animal cell, you will find that the one major difference from the outside is the presence of cell wall in plant cell, whereas it is absent in animal cell. So the isolated protoplasts are the source of functional organelles. They can be useful for studying various metabolic pathways, transport properties of distinct membrane. Then the question arises why it is needed. If you isolate a protoplast, it can be regenerated into complete plant. Protoplasts from same or different species can be fused to form hybrids. That is called as somatic hybridization. And these protoplasts are useful for in taking macromolecule like nucleic acid and proteins by process of phagocytosis. This is technique, that is chemical method, where protoplasts are surrounded with molecular foreign DNA, and with the help of fusion like polyethylene glycol and calcium ions, the porosity of protoplast changes and nucleic acid enter into the protoplast. Another method of electroporation in between two electrodes of protoplast is kept surrounded by nucleic acid or foreign DNA, and once the current is passed, the nucleic acid directly enter into the protoplast. So these are the two techniques of direct gene transfers. Basically, there are three methods of protoplast isolation, that is mechanical, enzymatic, and laser method. Mechanical method is suitable for those cells which are having large amount of vacuoles, and in which the cells are plasmolized and tissue are dissected with the help of fine blade, and they again deplasmolize. So this is a flow chart of mechanical method. Your plant cell get undergo plasmolysis by keeping in hypotonic solution. Then they observe under the microscope, and those cells which get plasmolized, they are cut with the help of blade, and by adding certain type of low salt solution, the protoplast gets swell and the release of the cell wall, and the protoplast gets collected. The advantage of this method is there is no need to use of hydrolytic enzymes, and components of membranes are remain intact, while the disadvantage includes the yield is very low and it is limited for only few tissues. Prior to go ahead, just try to look out the component of plant cell wall. Basically, it is made up of primary cell wall and middle lamella. Middle lamella is rich in pectin, whereas primary cell wall is rich in cellulose and hemicellulose. So, in enzymatic method, there are certain enzymes like pectinase, cellulase, hemicellulase, and mannitol. So, hemicellulase are used to degrade this pectin and cellulose and hemicellulase, whereas mannitol is used as an osmoregulant. It is a flowchart of enzymatic method where leaf is initially sterilized, whose lower epidermis is removed, and that is cut into pieces. Basically, they follow two pathways. The first one is a direct method in which the leaf tissue are incubated with pectinase and cellulase. These pectinase and cellulase break down pectin as well as cellulose to release the protoplast. In another method, the incubation with pectinase is the first step, where the pectinase is remove pectin and the cell gets separated, and later on these separated cells subjected to the cellulase so that the cell wall is completely degraded and its protoplast gets isolated. This is a schematic presentation where leaf is cut into pieces, then incubated with the enzyme. Later on, they wash with cell and protoplast washing buffer, and ultimately protoplast isolated by centrifugation. If you incubated them on specific medium, they will regenerate and completely plantlets are going to form. The advantage of this method is it can applicable to variety of tissue. Minimum osmotic shrinkages occur, cells remain intact and uninjured, and protoplast readily obtained. The disadvantage is while using enzymes, certain elicitors are produced during the breakdown of cellular matrix, which may elicit the cell. And meantime, if protease used, that is going to remove the upper moiety of protein from the plasma membrane. The third and most advanced as compared to earlier one is the laser microsurgery method, in which UV laser microbeam used to induce precisely localized submicrometric perforation in the cell wall. Here the cells undergo plasmolysis, and this plasmolysis cell treated with laser pulse for few nanoseconds, and again cell gets deplasmolized so that a protoplast will merge out from the pores. So basically protoplast isolation include protoplast culture include isolation, culture and fusion. You will find how enzymatic method is used. The leaf tissue is subject to enzymes. Later on, the protoplast is float on the sugar solution. Here you will find green color bodies in the protoplast. These are nothing but chloroplast, and you can find the yield, the amount of chloroplast generated. This is two protoplasts to about fuse. Once they fuse, they start divide, and once they start division, they start to form they form calli. And when they expose to light, they turn to green, and then there is a generation of certain emergence of shoots, and they allow to proliferate on suitable medium. Multiple tissues are each multiple is able to convert into complete plantlet. So there are wide applications of protoplast. Regeneration into whole plant, hybrid can be developed, easy to perform single cell cloning, genetic transformation, and excellent material for extracellular study.